The question is, why is it very important that trypsin is amplified in the inactive form? Okay. Enzyme is the inactive form of an enzyme. So trypsin exhibits this kind of property. So it is released in inactive form. The data has to be activated to the active form. So the inactive form is trypsinogen, and then the active form is trypsin. So the reason as to why it is very important that it is synthesized in an inactive form is to prevent the enzyme from digesting proteins in tissues or cells where it is. Okay. So let's look at one concept, and this concept relates to other molecules that are basically associated with the function of enzymes. So enzymes, they appear to be working alone so far in our description. But the truth of the matter is that they require other molecules, and these molecules can be organic molecules, or they can be inorganic molecules. So what do we call them? We have about 4 times that we need to get to know. So the first term, what is apoenzymes? Yes, we have looked at something that we have termed as zymogens. We looked at zymogens. We said that when enzymes are being produced, they are produced in inactive forms. A key term at zymogens, and we said that trypsin is one of those enzymes that exhibits that characteristic, that it's separated in an inactive form, and it has to be activated in the right place that it can carry out its function. Now, it's the same thing that we have here. When the concept of apoenzyme is basically the same thing, that we still have enzymes that have to be activated by what we call cofactors. So what is cofactors? Actually, these cofactors basically activate these enzymes that they can be able to work. So these cofactors can be organic in nature, or they can be inorganic. So the cofactors, we are saying here that they are inorganic components that enhance the activity of the enzymes. Okay, so cofactors are inorganic components. So the enzyme is so the cofactors are inorganic. So we can have magnesium ions, iron 2 ions. These are very important components that because they can enable the enzymes work better. So they are able to enable processes. For example, iron is very important, and magnesium are very important to processes such as energy production, chemical signaling, and many other processes. So, for example, also zinc is actually very abundant in the enzyme alcohol-aldehyde derivatives, and magnesium very important in the synthesis of ATP, especially in the enzyme called ATP synthase. Okay, and then the other components that are needed are the organic components, and these ones are the ones that we term as NAD, then FAD. These are coenzymes, they are organic in nature, and if you go back to the electron transport system of the electron transport system in respiration, you realize that these are actually important electron carriers and they help in energy production. They actually very important for enzymatic action. Okay, so that means that they play an important role in enhancing the activity of enzymes. Okay, and then the last term that we had is the protetic group or the protetic groups. These are 9 amino acid components that are tightly bound to the enzyme, and they are very important for enzymatic activity. So that means that, for example, heme is this kind of contains iron, is very, very important in hemoglobin function. So hemoglobin is a protein, but that hemoglobin has iron. and without that cannot or skip be transported. Okay, so the protected group is the non-amino acid component that are tightly bound to protect. It's a very important for their function. And remember that enzymes are also protein. Okay, so we go through the questions and see how we can relate those concepts to the questions that we have. So an enzyme was found to be inactive until magnesium was added, after which it catalyzed the reaction efficiently. So that means that the enzyme needed magnesium for it to work better. So in this case, what is the role of magnesium? How do we call magnesium? In this case, the magnesium is a cofactor, and then the enzyme part, which is proteinax, is actually termed as apoenzyme. Zymogens. Okay, zymogens are inactive forms of the what? Of the enzyme. Okay, so the zymogens have to be changed into active forms, right? Now, when you look at the protein, the enzyme has the protein portion that is non-active, and that is what we call the apoenzyme. The enzyme has an active part, the active site, and the non-active part. So the proteinous portion of the non-active part of the enzyme you would determine as the coenzyme. Okay, so when you have an enzyme was found to be inactive until magnesium was added, after which the reaction occur efficiently. And what is the protein-only portion of this enzyme? That is apoenzyme. Okay, and what name describes the magnesium in this context? It is a cofactor because magnesium is inorganic. Okay, the inorganic molecules that support enzymatic action are called cofactors. And then, what is the combining functional enzyme called? It is a holoenzyme. Okay, so we move to question number 2. A scientist study alcohol metabolism and finds out that actually zinc is very important for the enzyme alpha-proteohydroxylase when it makes to function. So in this case, what is zinc acting as? Cofactor. And then the other question is, why are cofactors get contact for enzymatic action? Because they help complete the enzyme active sites or participate in the reaction, enable catalytic action. And then state another example of a cofactor. So magnesium in ATP synthase, but you can also have iron in different other reactions. Okay, so you have in a cellular respiration NADH and is reduced to NADH to carry basically electrons from one reaction to another. What type of molecule is that? NAD, nicotinamide adenine dinucleotide. What is the role there? What type of molecule is it? Coenzymes, because it is organic. It's an organic entity. It acts as an electron carrier, transferring electrons during redox reactions. Okay, the last question. A mutation disrupts the attachment of human group to an enzyme, resulting in loss of its biological activity. What type of enzyme component is the heme? Heme. cyclopeptide group, the non-amino acid component. Yes, that tightly binds to the what? To the enzyme. The coenzyme is loosely attached but can be touched. So that heel is very important in transportation of oxygens